This is a production of Cornell University.
Okay. Next up today we have Kirby Peters. Kirby is from Roxbury, Connecticut. He worked with Matt Ryan, Jeff Liebert, and Sandra Wayman in soil and crop science, and his post-graduation plans are to be a greenhouse manager at Johnson's Backyard Garden in Austin, Texas. The title of Kirby's seminar is Soil Seed Banks on 4 Organic Grain Crop Farms in Central New York.
Okay. So hello everyone again, my name is Kirby Peters. I'm an agricultural science student graduating in a few weeks. And the title of my project that I'll be presenting to you today is the Soil Seed Banks on 4 Organic Grain Crop Farms in Central New York. And my thesis advisor again was Matthew Ryan. So first I'm going to give you a quick little outline of the project. What we heard from several farmers that we know is that red clover had been identified as a volunteer crop on several in several farms. And so what we decided to do was to sample the weed seed bank in order to identify if this was truly, if there was truly a red clover seed bank within the soil. So what we measured is the weed seed density, the weed species richness, the red clover density, and lastly, we looked at the weed seed community as a whole. So first, I think we should talk about what a soil weed seed bank is. And this is just a viable seed in the soil that has the potential to germinate. Red clover specifically is what we're looking at. And this is extremely common in organic agriculture, mainly because organic agriculture cannot use synthetic inputs. So organic growers tend to rely on biological inputs such as red clover, because it's a legume and it can fix nitrogen into the soil with its symbiotic relationship with bacteria. So as you can see here, here's a picture of red clover in case you didn't know. It's a nice drawing. And like I said, a few growers have mentioned that they have seen this crop as a volunteer crop. And what that means is basically the crop has persisted past its time in the rotation. So clover can be frost seeded or can also be seeded in the fall, go through the winter, and then reemerge in the spring. And that's great and all. It's then terminated in order to make room for the crop. That could be a grain, as we like to say, wheat. But the issue with a volunteer crop is then it persists past that, and then it's still present in your crop. So many people see this as a negative implication, because you have this red clover that's competing with your crop for whatever resource, whether it be nutrients, sunlight, or water. However, this study kind of takes it in perspective. This study looks at the possible benefit of this volunteer red clover. I mean, there's many benefits to red clover itself. Like I said, it fixes nitrogen. However, if we don't have to seed red clover, if there's just this like natural red clover seed bank, you can save seed costs. That's less labor. You don't have to make a trip into the field to broadcast. And you can also potentially control the emergence of your red clover. This is beneficial because there's times, for example, wet times of the year when you cannot get into your field because it's too wet. However, if you have this red clover seed bank, it'll come up anyways. So it's always a nice benefit. So in order to look at this red clover, what we did is we looked at it within the context of the whole weed seed bank. So from there, we decided just to make a characterization of the whole weed seed bank itself. So what we did is we did the study over 2 years. This occurred in 2015 and 2016. We sampled 4 farms. These farmers were chosen from our advisory board in our lab. These are pretty experienced organic growers. These are organic growers that were some of the first organic growers in the state. So when I say they're experienced, they're very experienced. And so we're thankful for them to volunteer for the study. What we did is we asked each farmer to pick 3 fields, their clean field, a high clover field, and a weedy field. The idea with this is when they chose, for example, a high clover field, it would be identifying a field with a red clover seed bank present in it. When farmers were choosing their clean fields, generally they were picking fields that they observed low weed abundance in, at least above ground, but also they talked about how conducive fields were for control. So one farmer mentioned the friability of the soil as making it more conducive for control. For high clover fields, farmers talked about basically the presence it had in their rotation, but also all the farmers were harvesting red clover. And for these fields, they said they noticed that a lot of seed was dropping down to the soil. adding some red clover seed into the soil seeds bank. And lastly, the weedy field. Farmers chose this generally because they had observed a high amount of abundance in these fields. And also they talked about how some of these fields were not so conducive for control. So some farmers talked about fields being really wet and that kind of preventing them from getting into the fields and controlling weeds. So here's just an example. This is the Potenza Farm. So you can see he chose his weedy field here, his high clover field, and lastly his clean field. And then from there, we did our soil sampling. And we split each field into 4 quadrants. And then we sampled each quadrant. Soil sampling was done in early spring before any weeds had emerged. As you can see here, there are no weeds present, no weeds here. And soil sampling was done with a soil probe. This is what you see here and here. We would go 8 inches down. The diameter of the probe was about 5/8 of an inch. And we did 30 soil cores per quadrant. And then from there, what we would do with the 30 soil cores is we would combine them in a bucket, as you see here, to make one composite sample per quadrant. So all in all, we had 4 farms, 3 fields, and 4 quadrants. That was about 48 samples for 1 year, and after 2 years, we had 96 samples. So once we had that composite sample, what we did is we performed a greenhouse germination bioassay. This is what you see here. We would take 1 kilogram of soil from each of those composite samples and put them into one of these trays. So each sample is represented in one of these trays. We would allow the weed seed in the soil to germinate. And then once we were able to identify, we would identify them, count them, and then remove them. And we would identify them by genus and species. So what we would do, this is just an example. So once these had germinated, we would then identify them, count them, and remove them. That's considered 1 flush, and we did 2 flushes. So after we identified, counted, and removed, we then stirred, mixed, watered to hopefully stimulate some more germination of weed species. And then again, let them germinate, identify them, remove them. So that's why we call it 2 flushes. In order to analyze this data, we use analysis of variance, or ANOVA. And then to compare the means, we use the Tukey-Kramer method. We analyzed, like I said in the beginning, weed seed density, which is just the total amount of weed seedlings we observed. Weed species richness, which is just the amount of species per farm or per field. Red clover density, which is just the amount of red clover seedlings we identified. And then lastly, the weed seed community, which is just looking at all the species and abundance in all. So first, what we're looking at here is the weed seed density we observed. You can see our response variable is the soil weed seed bank density of seedlings per meter squared. It's important to acknowledge that we compared within farms. So we just compared the weedy, high clover, and clean within each farm. The different letters indicate a significant difference. The NSC represents no significant difference. So the main takeaways from this is we saw a very large range of weed seed densities. So you can see in the Martins high clover field in 2015, right here, there was about 3,800 seedlings per meter squared identified. And then in 2016, in the Oshner high clover field, you can see it was much larger. It was about 103,000 seedlings. So this is a very large range. And then the next thing we took away from this was that almost all of the weedy fields had higher seed densities than the clean fields. So you can see here and here, except for the one example that we didn't see this was in the Meyer farm in 2015, where there was no significant difference between any of the fields. Next, we're looking at the weed species richness. It's important to acknowledge we're not looking in a area variable now. Now we're looking in a mass, so we're looking at species richness per species per kilogram. This was a little bit more variable. However, between years, it stayed fairly constant. So you can see here in Martins, the weedy field had a higher species richness than the clean field. In both years, for Meyer, it stayed consistent, no significant difference between any of the fields. Same thing with Oshner. You can see the weedy field, there was an observed difference between the weedy field and the clean field. And a little bit of variance we saw was between the Potenza Farm, where you saw the weedy field had a higher richness than the clean field. However, we didn't observe any difference in 2016. So lastly, we have the red clover density. There wasn't as much variation. You can see here, we didn't observe any difference in the Martins Farm between fields. Same thing with the Potenza. However, we did notice, we saw quite a bit of difference in the Meyer Farm, where in 2015 and 2016, the weedy fields had a higher red clover density than the clean fields. And same with the high clover. The only other difference we saw was in the Oshner Farm in 2015, where the weedy field had a higher red clover density than both the high clover and clean. So now we're looking at what we call the weed seed bank community data. This is just the species that make up the top 75% of the abundance from each farm. So as you can see here, we have the total amount of abundance of the total weed seed bank community. So what we noticed from this is that there was a lot of overlap of species between farms. You can see path rush here and path rush here. You can see fleabane species. Yeah, there's a few. Wood sorrel and other wood sorrel. There were some commonalities between a lot of farms. However, there were specific species that were common on all farms or the majority of farms. And some of these examples were purslane speedwell. You can see it made up a high percentage of martins in the Oshner Farm. It was a lower percentage on the Potenza Farm, however, it was still present there. The next example is giant foxtail, which we found on a lot of farms in very high percentages, which you can see in the Myers, Potenza, and Martins Farm. And lastly, red clover wasn't necessarily a common species amongst all farms. However, just for the sake of this project, we should be pointing it out. You can see in the Martins Farm, we had about 3% of the total abundance of weed seed was red clover. But what's really interesting, in Myers Farm, we had about 18% of the total weed seed bank density was red clover, which is a high amount of what we observed. So now we're looking at the 2016. Not much has changed. You can still see the similar common species. Giant foxtail, for example, is still present in these three farms. A huge increase, 43% of the seed bank is now giant foxtail. But yeah, once again, it's still present in the Martins and Potenza Farm. Red clover, you can see it's now has disappeared from the top 76% of the Martins Farm. It decreased by half in the Myers Farm, went from 18% to 9%. And now it is present in the Potenza Farm. So there's some variation there. So from some conclusions from this project, the first, we saw the majority of the weedy fields had a higher seed bank density than the clean fields, which suggests that there's some support behind the farmers' picking of each field. What's really interesting is we found the presence of red clover in the seed bank in many of the fields. So we found the red clover was present in 22 of the 24 fields over the 2 years. The density ranges from 0 to 4,000 seedlings per meter squared. And then lastly, we saw in certain farms that red clover had made up a large percentage of the weed seed bank. So for example, in the Myers Farm, it made up 18%. And then lastly, our last conclusion is that there were common species amongst the majority of farms or all farms. And those species were giant foxtail, pigweed species, wood sorrel species, purslane speedwell, common lamb's quarter, and common ragweed. And this adds to some research that we've already seen, where these species have been identified as common species in organic systems. So some next steps to take from this project would be to determine management practices that would support or promote the persistence of red clover seed banks. We know this is volunteering. We've also heard from many farmers that they're actually already utilizing this volunteer crop. So it's important that we start investigating other methods and ways of managing red clover. But also in the context of this study, it's important to explore differences between farms. So for example, in the Myers Farm, we saw very high, well, not very high. We saw high levels of red clover compared to other farms, and it would just be good to explore the differences in terms of management to see why they have such higher levels and how we could use those management practices in other farms. So this project would not be possible, of course, without the Sustainable Cropping Systems Lab, led by Matt Ryan, my great friend Jeff Liebert, Sandra Wayman, Chris Pelzer, and of course the 4 farmers that participate in this project, and then other people who just helped me with presentation, editing my thesis, and as well just other suggestions and helpful guiding lessons. Yeah. Thank you.
It's common practice not to use scientific names for the species you're dealing with.
Yeah, so I mean, I presented common names just because I thought it'd be easier for the just presentation's sake. But no, it is pretty common to use scientific names for species that you're identifying. Yeah.
So I don't know common names, because of that I'm wondering, are there any of those perennials?
Mm-hmm. Yeah, so we saw well, there was more annuals than perennials for sure, but there were some perennials. For example, wood sorrel was common. We found that at all farms, but at very low levels, about like 3%. Besides that, off the top of my head, I can't think of any that were common amongst all farms, but the majority were annuals. Yeah. Yeah.
One question is, I know you're planning to explore more differences between farms, so I wonder whether it has been done or you're planning to do that, to analyze soils more precisely, like you have seen, high humidity, so just to see whether there's something to do with mineral pH, mineral composition that could be advantageous. So this is one question.
Yep.
And second is regarding, so it looks that one of the farms, I think Maya's farm, so the red pole was initially like ranked number 2 by the abundance, but
Yep.
then it became clear to
Dropped off.
drop down.
Yep.
So I wonder whether, this nitrogen fixing ability provides benefits not just for crops, but actually it's taken advantage of by others.
Others, yeah.
By nature as well.
Yeah. So your first question, remind me.
Soil types.
Okay. Yes. So for the for the project for the paper at least that I'm writing, I didn't present it here, but we have taken soil health data from each farm, so that's something we can look at and analyze further. So this is for people who don't know, soil health tests are just comprehensive tests of biological factors, chemical factors, and physical factors. So soil structure, chemical, pH, and whatnot. So yeah, that. And then your second question, I'm sorry.
That's the advantage of having a clover for crops versus weeds.
Yeah, so it's hard with nitrogen. It's important to have a balance because there's plenty of studies that have shown that if you have excess nitrogen, weeds are way more competitive at taking up that nitrogen than crops. So it's really important. But in general, red clover as a biological source of nitrogen is fairly good at just staying available for crops, just because it's not immediately available necessarily because you incorporate it as a green manure. But that's just the gist I know. It's probably a little more complex than that. But
One more. History of each field is clearly a factor in what you see. How do you determine the history? How far back do you go? Did you core the field and look at it different times? And if you did, can you identify the taxa by the seasonal ethologies? Clearly that would have a dramatic effect on what you see in the feedback.
yeah, so I mean, it would be, I think a longer term study would be great. We only did 2 years, so we were only to see a certain time period, and there are a few studies suggest that the longer, it takes a while for seed banks basically to adjust to certain management practices. So unfortunately, we don't necessarily see that long-term effect of management because we only have the 2 years. However, we have gone back and we've gone about 7 to 4 years back of management practices from each farmer. So yeah, we didn't present it in this, but that is something we have been looking at. Does that answer your question? Okay.
Thank you, Kirby.
Yeah, of course.
This has been a production of Cornell University, on the web at cornell.edu.